welcome everybody from your coffee break. My name is Andrej. I'm an assistant professor in chemical engineering, and it's my pleasure to introduce our next speaker, Professor Ron Larson. So Ron Larson and I aren't in the same area. Most of my work in materials is in energy, and most of Ron's work is now in drug delivery. But I do appreciate the technical quality and the impact that Ron's work has had. And you don't have to take my word. In 2003, Ron was elected to the National Academy of Engineering for elucidating the flow of properties of complex fluids at the molecular level and continuum levels through theory and experiment. So Ron has pioneered the development of material design tools for controlling flow and transfer properties that are now in use by major companies such as Dow and Procter & Gamble for polymer processing, personal care products, and more recently, polymers for drug release. So today, Ron will speak on engineering hydrogels and microparticles for oral drug delivery. And he'll talk about multi-scale modeling of drug release as a powerful tool for rational design of pharmaceutical formulations. Please welcome Ron Larson.
Thanks, Andrej. I'm delighted to have been invited here to speak at this symposium, so I want to thank Rachel and the organizers. I think Michigan is a phenomenal place to do research in the area of materials, and I hope to show you how easy it is to get into new areas here. In fact, 6 years ago I really was not doing drug release at all. I was working in other areas of polymer science, and I'll give you these two examples to show you how I did get involved in this area, and how Michigan is such a great place to strike up collaborations and get going. So the two problems I'm going to talk about, one of them is drug release from implants. This will be an implant in the eye. Second one is drug release from oral delivery, from pharmaceutical pills. So the first problem, I got into this area because I'm in biointerfaces, the Biointerfaces Institute, that has fairly regular challenge grant programs where people who don't know each other at Michigan, haven't talked to each other, are brought together to strike up possible collaborations. In this case, the program had to do with diseases and treatment of diseases in the eye, which I knew nothing about, still don't know that much about actually, but it brought together folks from the Kellogg Eye Center along with people from medical school, engineering school, and so forth. And in the process, I met Professor Basirli at the Kellogg Eye Center, got talking to him, and pretty soon we had a collaboration going. And it was driven primarily by this outstanding graduate student, Maziar Mohammadi, who's passionate about having an impact on human health. And he helped bring in Professor Jordan Green from Johns Hopkins University in this collaboration. A couple of other graduate students were involved as well, Nisha Hollingsworth and Ying Liu. So the motivation here comes from issues of eye surgery, which are common, and afterwards you need antibiotics to prevent infection and steroids to prevent inflammation, reduce it. In addition, after a few days, pressure can build up in the eye, and so there's a risk of glaucoma, which then has to be treated with hypotensives. The standard treatment is eye drops, and these contain a number of problems. First of all, there are barriers to the drug getting into the interior of the eye, and you have your tear flow, which washes away most of the drug before it can cross those barriers. So the result is you have to over-administer. You have to use much more drug than is needed for the eye, and this risks inflammation and a reaction, allergic reaction. Also, there's a rigorous timing regimen that's difficult to adhere to. Every 4 or 8 hours, more eye drops, particularly for the elderly. So there's a tremendous problem with non-compliance using eye drops. So the idea here is to try to eliminate the need for the eye drops by putting a depot in the eye that would allow for drug release. So we're not the first ones to come up with this idea. There are a number of vehicles for releasing drugs in the eye that are in various stages of development. I list some of them here. Many of these, some of these at least, have a problem that they're not biodegradable, so surgery is required to remove the vehicle at some point. And others, none of them really release more than one drug at a time. So what our interest was is to develop a vehicle that can release multiple drugs, particularly 3 drugs, in a single vehicle. And the targets here were an anti-inflammatory agent, antibiotic, as well as an anti-glaucoma drug in the same vehicle. Each of these has a different time release profile that's desirable for therapeutic effects. We also wanted this to be injectable in liquid form, but form a gel-like form inside the eye to be a reservoir for drug release. So to do this, the design here was to develop a material that would be liquid at room temperature but would turn into a gel at body temperature. So the aims here were first to obtain this rheologically transforming material that goes from liquid to solid over the right temperature range. We needed to have adequate drug loading and release quantities on a daily basis according to the regimen required for each drug. And we wanted to be able to finely tune the drug release so to make this vehicle versatile so that other drugs could be released as well. So the basic strategy here is to use a hydrogel, and these are made out of tri-block polymers. The center block is always the same. It's PEG, polyethylene glycol. The end blocks are more hydrophobic and could be either PLGA, shown here, or PLCL. These are commercially available. They're FDA approved. And this gives us versatility to design a vehicle. These form the PEO block is in water. It's water soluble, and the hydrophobic blocks form little nodules that link this together into a thermally transformable gel. And to that we add microparticles, which are hydrophobic, composed of PLGA, and these can contain our drugs. So we can make different microparticles, each containing a different drug, and put multiple drugs into the same vehicle this way. First we have to design this to be injectable. So we want it to be a liquid at room temperature and turned to a gel at elevated temperature. And this can be done by tuning mixtures of the tri-block copolymers. So we have multiple mixtures to satisfy this kind of a phase diagram. This particular one I show here is a 6-to-1 blend of a couple of commercially available tri-block materials. Then we want to release 3 different drugs. The antibiotic we chose is moxifloxacin. It's a relatively water-soluble drug, and you have to release that over about 7 days to block infection. Then the anti-inflammatory we chose is dexamethasone, which is hydrophobic. You want a fraction of a microgram or so per day over about a 2-week period, so a longer period. And then finally, the anti-glaucoma drugs, levobunanol, which is also hydrophobic, and you want about a microgram of that per day over a month or so. That one actually, especially, you want it to be released later after surgery when the pressure begins to build in the eye. So we load the dexamethasone and the levobunanol in separate microparticles, and then those are loaded in the hydrogel, while the moxie will just be put into the hydrogel itself. So we use a standard method to make the microparticles. So this consists of mixing the drug in one solvent, DMSO, and the PLGA that will make the particles in another one, dichromethane, which are mixed together into an organic solution that's then stirred into an aqueous solution of polyvinyl alcohol. And this will phase separate into these droplets that are then dried out. Microparticles are harvested containing the drug. So this just shows you one example of them. There's some tuning involved. In this case, with the levo drug, we found it was more advantageous to enhance its hydrophobicity. We deprotonated it, so the deprotonated form made the best microparticles and also the highest loading. So shows the release of this. This is measured by taking the vehicle, exposing it to an aqueous solution, and then using HPLC, measuring the concentration as a function of time. And you can see that the deprotonated form can release much more drug. In fact, reaches our target release rate over a 30-day period. This deprotonation doesn't affect the release of the dexamethasone from other microparticles, so these are independently released from the same vehicle, from their own microparticles. Likewise, in this case, we show that the hydrogel doesn't affect the release of the levo. That's controlled entirely by the microparticles. So whether you have the gel present or just the microparticles, you get the same release of the levobutanol. However, the dexamethasone, its release is strongly affected by the presence of the hydrogel. So without the hydrogel, you get this burst release, very rapid release, too fast, and then you've lost most of it. But with the hydrogel present, you slow this down to the desired rate. So this is very fortuitous that this is regulated by the hydrogel. And so in this case, we believe the reason for this difference is that dexamethasone has many hydrogen bonding moieties, which we believe will bind to the hydrogel. So you have the rate is controlled by both the release from the microparticles and then the ability to be transported through the hydrogel, whereas the levobutanol is regulated entirely by the microparticles. Okay, so now the flexibility that we get from this is from 3 different modalities. We can control the type of polymer used to make the hydrogel. We have a lot of variety here because we can change composition. So we can tune the hydrogel significantly. We can also tune the chemical properties of the microparticles, and I'll show you exactly how that's done in the next slide by the composition of the polymer encapsulating the drug molecules, and then we can control the amount of microparticles of containing each drug that you add. So you have tremendous flexibility in regulating how much of each drug is released as a function of time. And this illustrates this flexibility here in the case of the microparticles. So those are regulated, these are PLGA, so you can regulate the amount, the ratio of the lactic to glycolic acid in this polymer, and that has a big effect on its hydrophobicity, and you can see the strong effect on the release rate here that you get by controlling that. Very interestingly, you can see this acceleration in the release. That's actually fortuitous as well, because you want this drug to be released later on after the surgery when ocular pressure can become an issue. And this is apparently due to the fact that as the hydrogel decomposes, these are biodegradable, releases acid groups, which then accelerates the degradation of the microparticles and then releases the drug faster. And so you can see by regulating the composition here, you can control this release rate of this particular drug, levobutanol. So here's our final product, and shows this set of releases. You get the antibiotic coming out as you want it in about 7 days, moxifloxacin. Then the next slower one is the dexamethasone. So that's for anti-inflammation. So that's coming out over 2 weeks, most of it, but it continues to come out afterwards. And then finally, the slowest one, you want this one to come out over a 30-day period, and that could be regulated as well. So this was a successful strategy, and now we're going into animal studies, and we believe this has potential for commercialization. There's a huge market. Cataract surgery is performed about 4 million times a year in the United States. So the volume here is enormous. So this we hope has a great potential for improved treatment following cataract surgery. So this technology that just got started only about 4 years ago. We got started on this, and now there's a pharma company that's interested in this in licensing this technology. So that's an illustration of how quick it is to go from complete ignorance to thinking about commercial impact, and it's because of the connections here in Michigan, the med school, the eye center, and the vehicles that we have here, such as President Biointerfaces, to get people talking who didn't know each other, and a couple of months later you're doing a project together. So I think that's very exciting for me personally. So the summary is we have an ocular drug delivery vehicle that's capable of releasing multiple drug molecules. It's an injectable form that gels. We can get adequate loading of the drugs we need, and it's got the flexibility that we can imagine trying to release other drugs that might have a different release profile. Okay, so now I'm going to turn to a second topic. This is a project that's about 6 years old, and this is a completely different sort of beast. This involves the interest of Dow Chemical, which funded us for 6 years to do a rational design of drug release from oral drug release. Involved a number of grad students and postdocs that I show here, as well as outstanding colleagues at Dow Chemical. Again, the connection to Michigan is really important. This project was launched because Dow Chemical picked out 11 universities that it wanted to do long-term research with across the country. University of Michigan was one of those 11. And so in this case, I simply got a call from somebody at Dow that said, we want you to put in a proposal for this area. Here's the area. Would you put in a proposal? How much money? 1.2 million. Okay, I'll put in a proposal. When's the proposal due? Next week. Okay. All right, so of course I scrambled that week and got it in, and I had questions about it, and they actually fed me back comments. You could make it better this way, that way. Send it in. A month later, okay, here's the funding. Let's get going. So this was very exciting. Again, having this connection to Michigan is really vital. It gives you the credibility. Their company's very interested in working with us here. So in this case, their interest was in trying to more rationally design drug delivery vehicles using computational tools. And this would connect somewhat to what Sharon is talking about. The future, we hope, is going to be computational design and not trial and error. The first project I talked to you about was mostly the Edisonian kind of approach. And here we're trying to do something more rational. So hydrophobic drugs there are many potent hydrophobic drugs that have limited bioavailability, meaning they will crystallize in your GI tract. They don't get into the bloodstream. So the way to enhance their bioavailability is to formulate them with an excipient which suppresses crystallization. And these are typically spray dried to make what's called a solid dispersion of crystallizable drug with a polymer. And then the idea is that the crystallization is slowed or suppressed by the presence of the polymer that somehow interacts with the drug and inhibits its ability to crystallize. So what Dow is interested in is how to actually design these more rationally than just the trial and error approach. So they wanted to understand and control the drug stabilization and optimize polymer composition, hopefully to do it on a drug-by-drug basis. So you invent a new drug, you design an excipient especially for that drug that will be effective in inhibiting its crystallization. So we want to do this computationally. The polymer that is used here is a derivative of cellulose, so it's obviously it's biocompatible. Cellulose monomers have 3 substituent positions on which you could attach different groups. And the groups that are FDA approved are a methyl group, hydroxypropyl, acetyl, and succinyl. And with these groups, you have enormous compositional variability. You have 3 substitution positions. You have these 5 substituents that can be combined in any ratio you want. So parameter space is absolutely enormous. And we want to use computer simulations to try to do the design. So we immediately run into a problem. If we use our typical atomistic simulations, we're simulating here oligomers of the polymer, up to 40 oligomers, which is still pretty short. The water is not shown in these simulations, but these are in water, and you can see these kind of aggregate structures that form at room temperature, and if you raise the temperature, you see some change, but not much. But we know experimentally that when you raise the temperature from room temperature to elevated temperature, these go from a clear solution to a gel. So you don't see a sign of that in these MD simulations, which are limited to about 15 nanometers cubic boxes. And you can't get much farther than this with atomistic simulations. So to address the problem here of the kind of gel that forms and how it encapsulates the drug, we need a coarser grain method. I should also remark that this is a micrograph of the gel that forms. And what you can see in the micrograph are these fibrils, which form the gel. And these fibrils are about 14 nanometers in diameter. These form elevated temperature. And the 14 nanometers is about the box size of the MD simulation. So we don't have a hope of using atomistic MD to predict the kinds of structures that are formed. So we have to coarse grain. So we do that in a systematic way by coarse graining each monomer into a single bead. But because the interactions of this monomer depend on its substitution pattern, for each substitution pattern we need a different bead and different bead parameters. The size and the interaction. So we have to get these coarse grain parameters somehow. So we get them from atomistic simulations, which give realistic interactions. So our tool of choice here is called the pair distribution function or radial distribution function. It's basically the local concentration around one monomer of another monomer of that or another polymer. And so the peak here will give you some indication of the strength of interactions. And we get those from an atomistic simulation, and then we tune the coarse grain parameters to match at least the first peak of that at both room temperature and then elevated temperature. So this is for a homopolymer made of the same monomer, in this case unsubstituted, which would be cellulose. And we can also do the same thing with monosubstituted, di, or tri-substituted. And you see the peak changing, and we then match those peaks. The dotted symbols are atomistic, and the lines are the coarse grain. So we match those by tuning the parameters to be able to capture the effective interactions between the monomers. There are 3 substitution patterns, so there's actually 8 different monomer types. I just showed 4 of them here, but we do that for all 8 of them. If you then simulate shorter molecules, oligomers, at the atomistic level and at the coarse grain level, you can see just by visually that the structure of these aggregates are comparable at the coarse grain level as they are at the atomistic level. And we can look in more detail by looking at a single polymer. Now we can look at a very long polymer that we can simulate at the coarse grain level and obtain the radius of gyration, essentially the coil size, as a function of the number of monomers going up to 1,000 monomers. And we find the coil size matches the experimental coil size, which is from these colored symbols here, indicating a correct simulation at the coarse grain level. This simulation is for the commercial product, which is a random copolymer of each of these 8 monomers with the methyl substitutions. So we have combinatorics gives us 8 different monomer types, and these are the percentages or fractions of each monomer type in the commercial product. And we assemble those, we use those percentages in our simulated polymer to match the experimental polymer. Okay, so now this is at room temperature, and we know at elevated temperature this polymer would form a gel. So if we raise the temperature, we see above a critical critical polymer size, we get a collapse. The polymer collapses from this coil into something else. So now we can look at what that is, and we find it collapses into a ring, interestingly enough. And if you drop the temperature, it opens up into a coil again, and this is reversible. We'll go back and forth between a ring and a coil structure. And this video gives you some idea of how that happens. So this is at 50 degrees C. This was after we had done the matching of parameters. We simply did the simulation at the elevated temperature, and we immediately see this interesting structure form. There are actually 3 polymers in this particular simulation. One of them rather quickly formed a ring. And you can see this other one hasn't formed a ring yet, but it's sort of duking it out with this ring here. And eventually, the ring basically templates the other polymer, which is sort of wound down like on a spool. It starts to wind itself up onto this ring, indicating that it wants to form a ring. There's another polymer here, this red one, has folded up. It's not in a ring, but it's about to be pulled into this ring. And then you can see that these two structures are going to have to duke it out to see which structure it wants to form in the end. And you can see they're kind of fighting it out here. It's kind of a melee there. And then eventually the ring wins. Okay, so this is what always happens with these. It's actually quite a delicate interaction that would form a ring. So then if we have more and more of these things, these rings will build up and they start interacting with each other like this. You can only do this with a coarse grain simulation. This is way too much time to simulate atomistically. I'm not going to put you through the entire video here, but you can see from these snapshots what eventually happens. These things begin to grow these little tubules, and so we believe if you had enough polymers in here, these would grow long tubules. The diameter is 15 nanometers. So this is in fact what these fibrils are. They're tubes of polymer that have wrapped up like this. So this is a starting point then, and now that's just with one substitution group. We also have other substituents like the hydroxypropyl acetate, succinate. And so we can do a similar procedure with those. We use atomistic simulations to get radial distribution functions. We match them to our coarse grain model. The succinate group is big enough that we need a separate bead to represent it. And then the combinatorics of this, of course, is enormous. With one substituent, you have 8 different monomers. Now we have, say, 5 substituents, and so you have impossibly many different monomer types. So we took the ones that are most prevalent in all the products. So these are present at greater than 1.5% level, and we ignored the others and just used these 10 monomer types and developed force fields, coarse grain force fields for those. And then we can simulate the homopolymers of those. And you can see, well, here are the atomistic ones, and then we can look at larger and larger coarse grain versions, and you see the different structures that form from these homopolymers. So this gives us a sense of what kind of a gel you get with each of these. Okay, so now we have to introduce the drug. So we'd like to simulate what happens, how the polymer interacts with the drug. We need a coarse grain model for the drug. So this is the one drug that we chose, called phenytoin. It has 3 phenyl rings. 2 of them are identical. So we choose to coarse grain it into just 3 beads. This is a crystallizable drug, and we want this coarse grain model to be able to form a crystal. So to do that, we need a complicated force field. So we have the atomistic crystal structure, which is shown here, which we can simulate atomistically. We then get bead to bead radial distribution functions for the cluster of atoms that represent each of the 3 beads. And in the crystal structure, you get this complicated multi-peaked radial distribution function because of the periodicity of the crystal. And now we need a force field that will generate the same crystal structure or the same radial distribution function. So we use a complicated multi-peaked radial distribution function that we get by a process called iterative Boltzmann inversion, where you have a target radial distribution function, which is from your atomistics, and then you have your radial distribution function from your coarse grain model, and then you iterate on your potential. So this is a little bit like what Sharon was talking about, where it's a design of a potential here, design of a potential for a coarse grain model that gives you the correct radial distribution function, and hopefully then would let the coarse grain model simulate correctly the crystal of the drug. And in fact, we succeed in this, and we get the correct crystal structure from the coarse grain model with the correct unit cell parameters. This is an example of the coarse grain model and the growth of a crystal here across a periodic box. Starting from a seed crystal, this will grow out to form a nice periodic crystal. This is the coarse grain crystal, and this is the atomistic one. And so now we can mix this drug with the polymer, and then it will form in water. It spontaneously forms aggregates, which are like our solid dispersion particles. And we can then begin to assess this quantitatively. How effectively does the polymer block the aggregation and eventually crystallization of the drug? So this one's very poor at blocking aggregation, whereas the one here on the left splits up the drug very effectively, and this would be an effective excipient at preventing drug aggregation. We're not able at this point to simulate de novo crystallization starting from nucleation. That's a very hard problem. Nucleation of even small molecules is challenging to do. We're working on that at present, but we can simulate suppression of aggregation by the polymer, and also we can look at growth of crystals as well. This shows release of drug from a solid dispersion particle. So here we're simulating one of these drug polymer composites. The drug is red here, and we can compute how much drug has gotten to a particular point, either a spherical shell or this surface here, and track that versus time. And we find this scales diffusively, as you might expect, so we can make smaller or bigger particles of this kind and get scaling of amount released that scales as the inverse, as a square of the radius of the particle. And this lets us scale up the drug release to whatever particle size we want. And then we find the effect of the polymer composition on drug release. So you find a very large effect of the particular composition of the polymer on drug release. And we want this actually to be slow, because if the drug comes out too fast, then it's prone to crystallize. So the excipient is designed to slow the release of the drug from the solid dispersion particle. And in fact, the ones we identify here as the most effective at suppressing, slowing the drug release are the ones that have been found empirically to be most effective excipients for phenytoin. So we think this is possibly a design tool that one could use to design these excipients. This shows the diffusivity that we obtain from our simulations, as well as the release time from a particle of a given size. And you can see this very large dependence on the composition of the excipient on the release time. And these correlate reasonably well with release times that are seen from mixing solid dispersions in aqueous medium in experiments. Finally, we're working on the issue of crystal growth. And so what's illustrated here is a solid dispersion that you start with here. There's water present as well. And then a crystal has been nucleated here on this boundary of the box, and it starts to grow. But it's suppressed by the presence of the polymer that you see mixing in here. The green and the red are the polymer, which slows the crystal growth. And so we can measure the crystal growth rate as a function of the type of polymer you have present. And also, this gives us an indication of how effective a particular polymer composition is at slowing crystal growth. What we're working on right now is to develop methods of computing nucleation of crystals. So where you have no crystal, you just have dispersed drug. How can we determine how fast a crystal will nucleate in the presence of polymer or in the absence of polymer? And for that we need acceleration methods such as metadynamics is one such method that lets you promote crystal growth and basically put a reaction coordinate on your system and drive the system along that reaction coordinate to determine rates. Okay, so I will summarize here then. First, I talked about formulation of a microparticle hydrogel release vehicle with promise for timed release of 3 different drugs in therapeutic quantities to the eye. And these can be tuned, the release profiles can be tuned through design of the tri-block copolymer hydrogel and through tuning of the microparticle hydrophobicity. And then we developed a computational model for polymer excipient drug interaction, which allows us to predict release of drug in the GI tract of orally delivered drugs. This is done through strategic coarse graining of atomistic simulations. This is done by using the atomistic simulations to develop a pair correlation function or a radial distribution function that's then used to tune the coarse grain model. And we looked at the effect of substituent chemistry on the release rate, found it to be consistent with experiments, and this suggests the development of a potentially useful design tool for polymer excipients. This has been, these computational products have been delivered to Dow Chemical for their use in their design. I thank you for your attention. I'd be glad to take some questions.